we're the company that does whatever it takes so that at some point in the future, you wake up and your new reality is that each day you're actually getting better. So you're feeling better. One definition of that is I feel younger, right? It's sort of if you're getting fitter or you're losing weight, right? It's that similar trajectory where you're sort of feeling better, but actually getting younger, that's your experience, which is the opposite experience at the moment. Like all of us, we've all been younger, and we have different ages right now, but we've been younger, and we look back at that as a positive, right? Like, wasn't it great to be younger than I was today? So I'd imagine you wake up in the future and you're on a trajectory towards being younger. So whatever it takes to make that a reality, that's the company will be.
Today's guest is Dr. Daniel Ives, founder and CEO of Shift Bioscience. Daniel holds a PhD from the University of Cambridge, where he self-taught computational biology to develop a new therapeutic strategy for a rare mitochondrial disease. In 2017, he founded Shift Bioscience with the goal of translating cutting-edge molecular biology into practical interventions for aging. Over the years, Shift's focus has moved from mitochondria to mouse epigenetic clocks to what may now represent a paradigm shift in longevity science, the development of AI-powered cell simulations to discover novel rejuvenation interventions. Did I get that right, Daniel?
Yeah, there's a lot of words in there that you have to. Yeah, that was correct. That was correct. Thank you.
I should also say that you recently raised 16 million to scale this approach, and in today's conversation, we'll trace the science and the strategy. And for the first time, well, when this airs, it will be public. But I guess you haven't talked to that many people outside your organizations about a new study that you have published. And it's really, really cool. And we're going to talk a lot about it today. But maybe we should just, first of all, I gave people a little bit of your background, but maybe you could fill in the gaps here and talk about where you come from and how you ended up doing what you're doing now.
Sure. So I come from Cambridge in the UK, just a small university town north of London, if you don't live in the UK. And I studied at university in London, originally, doing biochemistry, and I didn't. It's not that I had a particular aptitude for biochemistry. It was just I was worse at everything else. So this was the one thing that I was good at for whatever reason. And around the time I was graduating, Aubrey de Grey had just given this quite sort of infamous TED talk where he basically said, "Look, now's the time to do something about aging. Like now's the time. The technology's there. Um, we should be doing something right now. We shouldn't wait another 5 years." And I think that particular TED talk captured a whole generation of scientists. It seems to be a common reference point. Like that's the talk people watched and they got involved. So I think after watching that talk, I was just compelled to find a project that addressed one of the what would become the hallmarks of aging. Aubrey described them as strategies for engineered negligible senescence, which is another mouthful. And at the top of the list was the mitochondria. So it's this particularly interesting organelle that produces your energy. The cliche is powerhouse of the cell, has its own genome that gets damaged with age. And Aubrey wanted to transplant that tiny little genome into the nucleus. And it was quite an ambitious sort of technical challenge. And I thought, well, if that's the hardest problem, why don't I have a go? Because when you're young, right, you can sort of take those types of reckless risks. So I basically reached out to a scientist in Cambridge who was getting sponsored by Aubrey to try and achieve this goal. And I said, look, I want to help with this project. And at the time, it was Mark Hammerlainen, who's now heading up LBF, who was actually the the postdoc on the project, or perhaps PhD student, they'd encountered technical difficulties and actually discontinued the project. I didn't have a plan B, so I saw there was a side project in the same lab that sought to achieve the same goal, which is basically get rid of this damage that accumulates. And that's where I got my start. So I basically started a PhD working on this. It was part of the University of Cambridge, um, and it went well, and that's how I got my start in the field.
But that's not what you're focusing on right now. Okay. What is Shift doing?
Correct. So in my PhD, I really believed, as a young naive person, that what I was working on was the cause of aging. And if I solved this problem, I would solve aging in its entirety. And so when I did, in fact, solve that micro problem in my PhD, I believed I'd solved aging. Like just imagine you were naive and you that sort of dopamine hit. It's like wow, because I remember that on the instrument there's a certain like signature. You can actually see the shape of the readout from this machine, and I knew exactly what that shape would look like. If it was the result I was looking for, and your heart is sort of thumping, right? You can hear it, and the result came in, and I really believed I'd done it, um, and at that point, I was like, look, we've got to make this real. Like, we've just got this to work in the dish. We've got to make this into a drug. And that was the beginning of the divergence from, my boss, who was the principal investigator of the lab, and what I wanted to do, which was solve aging. So the lab was focused on rare diseases caused by damage to this genome. I wanted to use the same approach to solve aging. So eventually, it came to a head because I wanted to do a very risky mouse experiment, and my supervisor didn't want to do it. The lab had basically failed to renew its grant, and we'd have to wait 6 months for the grant to come in. So I was offered redundancy or continuing on at half pay. So for me, the logical choice was take the redundancy, which is tax-free money, like so you get a nice lump sum of money, and pay a contract research lab to do the experiments I wanted to do, and that's exactly what happened. So I sort of came out, took the redundancy, used all that money to do the experiments I wanted to do, and I got the results that I wanted to get. And then that's the dangerous thing, right? Where you get the results you want at the beginning. It's a bit like gambling, right? When you win on the first coin, so you're like, oh, this is easy. so, so I'll just ask everybody else for their money and then do the same, right? So, but then it continued to work like that, right? Like, so, I'd ask for other people's money and then we get the next result. And, we were only using very small amounts of money. I think my first experiment was £4,000, which sort of for wet lab science is relatively cheap, right? If you think about all the infrastructure and containment and stuff that needs to be paid for, it's quite cheap. So I roped in my friends, I roped in family, they put in money. I got donations from a few individuals, and eventually asked my girlfriend at the time, I need to do the next experiment. Will you give me your life savings? Which is really unfair, right? Like at the time, I didn't even see how unfair that was, but she said, no, you should go to an investor that can afford to lose it, which is incredibly wise. And so, we basically went and talked to lots of different investors, all of which said this is just a crazy early science project. It's got no prospect of return on investment, and that was accurate at the time. But some of these investors looked kindly upon us, and eventually we were connected with an individual in Cambridge called Jonathan Milner, who's one of very few individuals in the UK that's created a life science unicorn. It was called Abcam. It's basically, scientists use antibodies to visualize proteins. And so he was like the vendor, like this website was the vendor for those antibodies. So he became an angel investor, quite prolific in Cambridge. He's got his fingerprints over many very interesting Cambridge companies. And we went to pitch to him, and he said, look, this is even too early for me, but I'm going to donate a small amount of money. The condition being that if this generates any results, I want first refusal on investment. And we generated the results, and he ended up putting in the first check. And just to answer your question, like why are we doing what we're doing now? The first thing that happened with that investment was we disproved the hypothesis behind the company. So, you know, the idea was we could solve aging by getting rid of this damage, and Beyond the investment, Jonathan was incredibly helpful because he had quite a broad scope of what was going on in the scientific field in general, not just in the narrow microcosm, which is mitochondrial biology. And he said, "Have you heard of the epigenetic aging clock?" And my response was, "What is the epigenetic aging clock?" And this was 2017, so 4 years after Steve Horvath discovered this clock. The moment I read up about this clock, I was hugely excited because this gave me the opportunity to prove to the world that my hypothesis was in fact correct. Like this was the dominant cause of aging, and I was going to use the clock to prove to the world that it was the cause of aging. And I did the exact opposite. We ran these experiments and we proved, well, to ourselves. We didn't tell the world, right? We didn't broadcast the fact that we were wrong, but we proved to ourselves that this wasn't the underlying driver of aging, at least what the clock represents, which is quite, it's the best signature, the best biomarker of aging that we have. It's not perfect. It's very easy to critique why it's not perfect, but it's so much better than not having an epigenetic aging clock. It's like having a compass, where previously you didn't have a compass. So the clock just didn't respond to this type of damage, didn't respond to the interventions we developed to reverse this damage. And so we ended up pivoting the company in the direction of clocks. So we asked the question, what's the ultimate use of a clock? And the ultimate use of a clock is to scan all of the biology, so systematically go through every single gene in the genome and assign a relationship to the aging process and let the biology tell us where a therapeutic should be targeted. And we basically set out to develop that type of clock, and it's basically a single cell aging clock was what we needed to develop to do that type of, very large experiment. And that's basically the underlying technology. You can sort of, if you go all the way to where we are today, that technology we developed, the single cell clock, compounds with something we call a virtual cell. So this is like enough of a representation of a real cell such that we can do rejuvenation experiments, but at high throughput. So just the scale of experiments you can do in a computational version of a cell is, in the it's orders of magnitude more than you could do in a wet lab. So my best description for this is that it's like putting your hand through a wormhole into the future, 2 to 3 centuries into the future, and picking out the best results and bringing them back into the lab, and that's the experiment you get to do, and that's the amount of throughput that we have with these simulations. And the amazing thing is that it works. So you take this simulated result, you put it in the wet lab, and we find these genes that we would never have found without the simulations, and they go beyond the Yamanaka factors. So a lot of this stuff is, they're tools that are only been used for the first time, so we didn't even know if they were going to work. So a lot of things coming together, but ultimately the proof is in the pudding. Do we find something that works in real cells, and now it's starting to work in multiple cell types, and we do. So that's why we're now excited about these tools, because they're actually delivering new biology.
Yeah. Should we maybe just give viewers and listeners just a brief explanation of, because we're going to talk about lots about partial reprogramming today. So maybe just start with, what is an aging clock?
Yeah, an aging clock is a molecular signature that correlates very closely with age. So it allows you to look at an unknown cell and say, what is the age of that cell, without actually knowing, what is the birth date of that cell. So, um, there's been a search for such an aging biomarker for a long time. I think there was a big NIH project that ultimately failed. But then, sort of in the 2000s and the early 2010s, Steve Horvath, who was originally looking for a biomarker of homosexuality, discovered a biomarker of aging in the methylome. So the methylome is basically the pattern of DNA methylation. So certain base pairs in DNA, you can add a methyl group. So it's basically a methane, and this basically influences the expression of genes. And so there's you can sort of look at the whole bulk of DNA and just look at where methylation is happening, where it's not happening. And there's a certain a set of methylation sites that behave almost almost with perfect predictability. They vary with age in an entirely predictable way across multiple tissues and now across multiple mammalian species. So there's some regularity in aging which can be used for us to understand how old a cell is. And this is basically called the DNA methylation aging clock.
And there are several clocks now being developed since the Horvath clock. What are their similarities and differences?
That's correct. So there's like a whole ecosystem of clocks that are developing, and each clock has its own specialism. I think that's the best way of putting it. So the original clock was it was such a breakthrough because it was multiple tissues. So it's called the multi-tissue clock. And before that, there were single cell type clocks or single tissue clocks, but people were worried that you were just like picking up an tissue specific artifact that would never apply to other tissues. So the fact there was a multi-tissue clock sort of gave you confidence. This represented the real aging process, not something artificial. So Horvath's original clock, it works in lots of tissues across the body, including the blood. But um, it wasn't very accurate for fibroblasts, which were basically the primary experimental system of scientists. So fibroblasts are a type of cell that you can culture relatively easy in a dish, and you can do different experiments. You can try different drugs. You can overexpress genes. You can knock down genes. Like that's the workhorse of scientists. And um, Steve Horvath and a scientist called Ken Raj in the UK subsequently developed something called the skin and blood clock, which basically adds additional fibroblast samples, fibroblasts of different ages. You add that to the training data set for these clocks, and you basically create a clock that's now more accurate in fibroblasts. And now, that's, for instance, that's the clock that we rely upon the most, this skin and blood clock. There's, if we sort of open this up a bit more, there's a lot of blood-based clocks um, that people have heard about, like Grim Age, Dunedin Pace Age, and what's important about these clocks and what's lacking from the multi-tissue clock and the skin and blood clock is um, sort of mortality and relationship to disease, relationships to sort of some of the things people suffer with. So, the original clocks were basically trained, like multi-tissue and skin and blood, were designed to be very good chronological age predictors. So it's the same as like looking at your watch, right? They were designed to predict the chronological age of the sample. But what humans care about, whole humans, is what's my risk of heart disease? What's my risk of when am I going to die? What's that date in the future? I guess not everybody wants to know that, right? But maybe it's just something that's useful information if you do want to know. And so these clocks like Grim Age and Dunedin Pace Age, they give you a much better connection to sort of disease, clinical markers, things like that. So they specialized in that facet of biology. But the amazing thing about DNA methylation, and the clocks sort of reflect this, but it's not the whole story. DNA methylation, for some reason, it's part of the cell. It's like a molecular layer in the cell or a molecular signature that can hear everything that's going on in biology. So it's so sensitive to what's going on that you can predict the number of packs of cigarettes that someone smoked from their DNA methylation. So it should be no surprise that this is the biology that can hear aging where no other biology gives you a good signature of aging. It's so sensitive to everything that's going on in the body. So the downside of this type of approach is that DNA methylation doesn't tell you why the body is aging. It's just these different sites are varying with age. It doesn't tell you how. It just gives you a way to measure where you are in a process, but it doesn't tell you the underlying biology. So they're not perfect, but you can't deny their utility unless you're pedantic.
I guess people use them also. I mean, you can buy these types of tests, commercial tests, where if you want to know your epigenetic age, but they're not that reliable, I presume, for individuals.
Yeah, so Steve Horvath, the the godfather of the clocks, he actually has he puts this very well. He says um, the use of these clocks for individuals, like on a consumer basis, is is mostly entertainment at this stage. So there's a lot of variability between samples.
Yeah.
Even within 24 hours, right? If you take multiple samples within the day, they can vary quite substantially. There seems to be some circadian, there seems to be some link to just circadian rhythms. But where they are useful is at a sort of group level. So if you're considering an intervention across a group, or a population, you can start generating quite meaningful conclusions. So they're quite useful at the group level from an investigator perspective or perhaps a healthcare payer perspective. But individual use is quite problematic at the moment. Um, Obviously, you can counter that by taking multiple samples and then taking the mean of those, but then that just puts the costs up, and I don't think um, there's not many people with a surplus of money that are going to fund the necessary number of tests to draw meaningful conclusions.
Yeah. You met Steve Horvath, right? You had a pivotal meeting with him. Can you retell that story you told me another time?
Yeah, that's correct. So after we disproved the hypothesis behind the company, um, that's a pretty serious situation in a company where, like, look, we just set up the company and the whole approach is flawed. And so I needed to find a new direction for the company fast, and I didn't want to leave any stone unturned. So I relocated from Cambridge to Silicon Valley because that was, at the time in 2018, that was sort of the center of gravity for the field. Like most of the science was being done there, most of the investment for this science was coming from that part of the world. And I basically was there to find a new direction for the company. I tried I had lots of experiments about where we could take it, and most of them failed. And the pivotal meeting was a workshop at the X Prize Foundation in Los Angeles. So I was very fortunate to be invited to this workshop, along with, Aubrey was there, Laura Deming was there, like all sorts of people were there that I had no right to be amongst. And the first topic of discussion was we want to run a prize for longevity, right? They wanted to do an X Prize for longevity, and fast forward to today, right? There's the X Prize health span, right? So that this was very early on, sort of in the genesis. And the first topic of discussion was we need a biomarker of aging if we're going to run a competition. And I'd just been using a biomarker with quite a big degree of faith because the clocks weren't popular at that time. They were they were black box technologies that made a lot of people comfortable. But we really needed to find the biology, so we were willing to take the risk by accepting the utility of these clocks. So I'd been using one. So I put up my hand and I said, there is a biomarker of aging. It's called Horvath's clock. Steve Horvath discovered this clock, back in 2013. He's in Los Angeles. Maybe we should invite him here, given we're all here. And what I didn't realize is he was in the room, and he put his hand up and he said, "I am Steve Horvath." And and I was like, that's fantastic, Steve. You should take over the discussion at this point. And so he did, and the conversation became very educated. And so in the break, I caught up with Steve, and he said, fantastic, you had such faith in the clocks. Shame about the results, but um, the faith in the clocks is sort of commendable. If you're trying to find the biology of aging, you should talk to my favorite collaborator, Ken Raj, who's back in the UK, who is the only scientist in the world actually trying to reverse engineer the clocks. Because that's the interesting question. Once you have this accurate biomarker, what's driving it? Like, can we get under the bonnet of what's going on? So Ken Raj was basically nestled in Public Health England, which is sort of not particularly known for its longevity biology. And I think Ken was using it to study the effect of radiation on the health of cells, and he was using the clock, like, does radiation affect does it affect the age? So I ended up like a boomerang going back to the UK and talking to Ken, and then basically finding our new direction by multiple discussions with Ken, eventually concluding with a single cell aging clock, which we developed, and that took us on this new path, which I sort of described briefly.
Mm. Yeah, maybe we should just give a brief brief crash course in Yamanaka factors and partial reprogramming as well. I think after that we have the foundation to move forward with where your company is today.
Yeah, happy to oblige, though I know so much about the factors and I'm not the authority on the factors, but I'll tell you what my understanding is. So, in the mid 2000s, a Japanese scientist called Shinya Yamanaka discovered that four transcription factors, which are a type of protein that basically is the manager of other proteins, they influence which other proteins get expressed. So these four transcription factors, when put together and overexpressed, can convert an adult cell like your skin cell back into a stem cell. And these they're called induced pluripotent stem cells. And to all extents and purposes, they resemble embryonic stem cells. So the first cell of life, right? After a sperm meets an egg, right? You have this embryonic stem cell. So it was a sort of remarkable discovery at the time that you could take a cell back through development to an embryonic stem cell. Um, and Shinya Yamanaka won the Nobel Prize for this finding, as did a scientist in Cambridge called John Gurdon, who'd basically done the same thing, but by taking an adult nucleus and putting it into sort of an egg cytoplasm. It's sort of a different way to reprogram. Fast forward 10 years after the discovery of Yamanaka factors, a scientist at University of Edinburgh called Tamir Chandra reanalyzed Shinya Yamanaka's data um, with the epigenetic aging clock. So Shinya Yamanaka had collected gene expression and DNA methylation data across a time course where fibroblasts are transitioning into embryonic stem cells. And he found that the epigenetic clock reversed all the way back to zero with this transition to embryonic stem cells. So at the time, it was exciting if you could slow down aging, let alone stop aging. Reversal was beyond the realm of the possible. So the fact you could reverse aging was just this spectacular moment. I'd say the second breakthrough in the field, the clocks being the first one, but then having this robust biomarker and showing it reversed was, sort of opened people's eyes. So the time this was published was just after I'd disproven the hypothesis behind Shift. So not only had I disproven my hypothesis, somebody had moved the goalpost and said we can actually reverse aging. And so the opportunity then became what's wrong with these Yamanaka factors. And they are incredible. They can reverse the age of cells, and there's a certain amount of safety you can eke out of these factors, but they were always designed to turn adult cells into stem cells. That's what they were optimized for by Shinya Yamanaka. That's what he was trying to find, and all of his experiments were designed to optimize for that activity. They were never designed to rejuvenate, and so you have this carryover activity, which is they turn adult cells into stem cells. And when you do this in an animal, you create these multi-tissue tumors called teratomas. So you basically create a stem cell in an animal, and that stem cell has the freedom to become whatever it wants. So you basically have this tumor risk tied up with this particular set of four genes.
Mm. And yet, I mean, yet we have all these companies, Altos Labs, or Retro, and Youth Bio, and we have all these companies using these Yamanaka factors in a partial fashion, right? So in a cyclical fashion, just turn back the time a little bit. But what you're saying is that maybe the Yamanaka factors isn't the way to go here.
Yes, so as a scientist, sort of looking at the Yamanaka factors, your immediate question is how are they rejuvenating? And the amount of information we have at the moment is precisely zero. So, we have these factors, but that's the whole story of rejuvenation. We just know they exist. And we also know they were optimized to turn adult cells into stem cells. That's a huge question mark, which is absolutely ripe for scientific discovery. Um, and, and at that point, you're like, well, why don't why don't the scientists get to work? Well, how do we find something that's better than a Yamanaka factor? We have to have the tools to directly ask the question, how can we rejuvenate a cell? So do we have the tools that are capable of allowing us to ask that question? And we need to be able to ask that question lots of times because there's four Yamanaka factors. So if I was just to test, I think it's all combinations of three transcription factors, which is sort of the minimal subset of Yamanaka factors required for rejuvenation. I think it's 562 million combinations, which is just unfeasible. So can we create tools that allow us to meaningfully explore alternatives to Yamanaka factors? And the machinery or the tools was just not there. Once this finding came in, the tools weren't there. So people just, they just optimized what was known rather than struggle with creating the tools are necessary to go beyond the factors, and that has been a big struggle, and that's basically what we decided to do. It's a bit of a snobbish position, which is the Yamanaka factors aren't good enough. What if rejuvenation can be separated from pluripotency? What if we could just ask the question, can we rejuvenate without asking the question, without also asking the question, can we be pluripotent? So could we create those tools? And the good answer is you can create those tools. And once you start using those tools, it turns out you can move beyond the Yamanaka factors. But that was the critical thing. Like, how do we create tools that allow us to go beyond the factors?
So what tools did Yamanaka use? I mean, he did not have these tools, but how did he succeed in identifying these factors?
Yeah, so Shinya Yamanaka, he he had the benefit of lots of genes that had already been identified as expressed only in stem cells. So yeah, there were lots of genes that were only expressed in stem cells, and so if you're trying to find a way to turn something into a stem cell, you can use that one of those genes as a marker. So what he did was he attached an antibiotic resistance cassette to the tail end of one of these genes, such that if you were able to create a stem cell via your experiment, the resulting cell would be resistant to an antibiotic. So when you apply an antibiotic, only cells that become stem cells would survive. So he basically just pummeled a ton of cells with lots of different hypotheses. Basically, it was like a bucket of 24 genes together that he had cause to believe might be able to achieve, um, you know, basically converting an adult cell into a stem cell. So, and then he applied the antibiotic and he saw that some cells survived. And then he basically went through systematically those 24 genes, narrowing it down to a minimal subset of four where the cells still survived with the four genes. But he had the advantage that he could basically select for the outcome that he wanted. Um, we haven't been able to identify up until this point a single gene that represents rejuvenation. It just hasn't been, like there isn't that definition. So we can't create the same system. So it's a more complex task where we need to basically find a definition of aging and be able to, run experiments with that definition of aging. But Yamanaka had this head start where he had basically a one-dimensional definition of pluripotency, which is what he was optimizing for. We don't have a one-dimensional definition of aging, and we still don't have one, but we have a way to measure aging now, which is high throughput.
Do you remember when you had the idea that, I mean, the Yamanaka factors is a dogma in this world that you're supposed to use the Yamanaka factors when you're doing partial reprogramming. Do you remember when you had this idea that what if we could use something else?
It wasn't my idea, so I'll just start with that. So this I'm basically going to talk about what has now become a redundant technology, but ultimately it did lead us to where we are now. So um, when we created this single cell aging clock, we didn't use DNA methylation sites as the basis of the clock. We used genes, and this is very important because we know a lot about genes in a way we don't know about these individual methylation sites. So if you create a aging clock that's built out of genes, you then start looking at the genes and you're like, wait a minute, if this is such a good aging clock, then these genes must be linked to aging, right? Like it's sort of, it sort of sounds obvious, but we didn't even think of this to begin with. We just created this clock, and we were going to like do a big screen. But then we started looking at these genes, and we noticed there was known aging biology sitting inside our clock, and it sort of validated that the clock was something. It was like externally validated. It wasn't just us creating some random clock. It's like it seemed to link up with known biology. And so then we looked at what's the best gene in the clock, or like which gene is has the greatest weighting in a clock. So a clock is usually like 100 different genes, and they each have different weightings and contribute to the clock. And then one gene will be more heavily weighted. So we then said, well, the genes with the highest weight, they must be really important in aging biology. And many of these genes hadn't been linked to aging previously. So we then said, well, surely they would be really good targets, like drug development targets. And so we ended up defining our clock as a driver clock. So, we basically look inside the clock to find gene targets instead of using the clock in a big screen to find the gene targets, mostly because it was much cheaper for us to do so. And I was trying to pivot a company. So you don't have a lot of money or time left. So money is always on your mind. So we ended up, looking inside the clock and sort of finding potential gene targets, and that was the spark. It was like, look, we could find something here that's specifically designed to target aging. It's not tied up with pluripotency, right? Like we might have biology here that's represents improved factors. It turns out we actually found uh, more pluripotency biology. So that biology that we thought was driving aging was actually pluripotent biology, which is sort of a hilarious turn of events. And we're still scratching our head over that one. But this was sort of one of the failed paths along the way to where we got to, which was basically using the single cell clock as originally intended, which was to do screens, but to then do screens in virtual cells. And that's how we found the biology that's now proving to be, I guess, more interesting therapeutically than Yamanaka factors.
Yeah, maybe we should talk about that now. So tell me, what are your findings? What does your study tell us?
So um, the stories, I think the story is interesting because we asked the question, how hard could it be to find new factors? And the answer to that was, well, it could be 562 million combinations. How are we going to meaningfully explore that space? We can't do this in the wet lab. It would take, centuries. So therefore, we need a virtual cell. So let's get a virtual cell. So we were all geared up, right, for this Everest climb. Like we're going to explore all of this, and
Maybe we should, Daniel, maybe we should just, I mean, I think when people hear virtual cell, they hear different things. What is a virtual cell to you specifically?
so for our purposes, a virtual cell is simply um, a representation of how genes interact with each other. So we're looking specifically at genes. We're not looking at any other part of the cell. So we're just looking at genes, and the same machine learning that was used to create ChatGPT, which is this transformer model, can basically be applied to look at the gene expression of lots of cells. And I think basically you can look at individual cells now with single cell gene expression. And looking at enough cells, these models can say, look, if this gene is up, these two genes are down. If these 15 genes are up, these 11 are down. It can stitch together how the genes are interrelated, and then you can basically take that like network or map, put that in a computer, and now you can in the computer say, if I upregulate this gene, what's the impact on all of the other genes? So that's what I mean by a virtual cell. It's just a very narrow representation of the cell, but it's a meaningful representation, and it's basically that's what we need. That's we just need to know what's going on with gene expression to determine if a cell got older or younger. So that's what I mean by a
Yeah.
virtual cell. So the stage is set, right? We've got a way to say, did a cell get older or younger? We've got a virtual cell, and that means we can run all the experiments, and we can say, did the simulation get older or younger? The first thing we discover is that there are multiple combinations of genes that are predicted to be rejuvenative, but they share a single gene. So this single gene keeps occurring in different combinations that are rejuvenative. And then we said, we basically said, surely not. Surely there isn't a single gene that's sufficient for rejuvenation because somebody else should have found it if it was that easy, right? It doesn't require a virtual cell. You could do, much more conventional experiments. Um, but nonetheless, we entertain the possibility, how easy could it be? This is always the fun one, right? Where it's like, you expect something to be hard and it could be easier. So we say, oh, how easy could it be? Let's just test it. Like, let's just speculatively test it. And it was rejuvenative on its own. And it's, comparably rejuvenative to OSK, which is sort of the subset of Yamanaka factors that are, I guess, safer. We have data that shows they're not absolutely safe, but certainly safer than all four Yamanaka factors. So we find a single gene, comparably rejuvenative, but doesn't induce pluripotency. So you don't see this pathway that sort of gives people cause for concern in terms of therapeutics. So yeah, two things there. Firstly, single gene, right? Like that was the reduction in complexity is enormous from a drug development perspective. Like going from three genes to single gene, that becomes a serious conversation with people that aren't in this particular field. Like sort of drug development people that would otherwise see this as very sort of make believe. And then the fact there was no pluripotency coming along, that was amazing because it's like we not only have we reduced complexity, we've removed this, basically this side effect that everybody tries to ignore in different ways, right? But that was a really big deal. And then again, we asked the question, how easy could it be? We were like, well, maybe, just maybe, the single gene might work in multiple cell types. But it's also a long shot because surely it couldn't be that easy, right? Single gene, no pluripotency, it works in multiple cell types. Because we geared up for multiple genes, only works in single cell types, long road towards getting rid of the safety problem. So we just tested this gene in another cell type and it works. And it actually works better in the second cell type, which is a blessing and a curse because it does show you can bridge multiple cell types, but it also shows you that rejuvenation is variable between cell types. So it might be less efficacious in a third cell type. So obviously the next thing we're doing is testing as many cell types as possible. But the big surprise from all of this is we geared up for this ascent to the summit of Everest, potentially dying along the way, and we found that it's a lot easier if you have the tools to just ask the question, like what is rejuvenative and what isn't rejuvenative at high throughput. So just having the tools allowed us to find stuff that was a lot closer than we thought it was.
So you identify this in your model, your simulation, and then immediately you try it out outside in the real world.
Correct. Yeah. Correct. So this is, a simulation is always exactly that, right? It's a representation, but never an absolute or perfect representation of the real cell. So um, we have to sanity check anything that happens in a simulation. It's like a Formula One car, right? You can build a car in a simulation, but you've got to put it on the track and see if you have in fact designed a better car or not. It's the same with your experiments. You do them in simulation, bring them to the real world, and they rejuvenate real fibroblasts without inducing pluripotency, and they rejuvenate a second cell type, which is keratinocytes, which is another cell type in the skin, but from a different germ layer, which is basically in early development. There's three germ layers. You separate into three different sort of types of cell, and we want to sample cell types across those layers just to prove to ourselves that we could tackle any cell type in the future. You're like, sort of diversity of cell type won't be an issue. So yeah, so that's yeah, we're still a little bit taken aback by the fact the genes exist.
But have you also tested them on whole organisms, like model animals?
Yeah, so we're currently halfway through a mouse experiment where we're delivering this gene by adenovirus vector to different organs in a mouse, just asking a very narrow question, which is can we express the gene in those organs, right? That's sort of the first thing before you get excited about whether those genes do anything. And if they do express, we're also going to just measure the epigenetic aging clock in those tissues. It's a very narrow experiment because we just wanted to go quickly. But yeah, that's the next question. Can we do this in something more like us, not just cells in a dish?
What do we know about this gene, and why hasn't it been discovered before?
So, as is the case for many genes in the genome, especially transcription factors, relatively little known about this transcription factor. There's so much I can say and there's so much I can't say, but what I can say is this gene is expressed in very limited parts of the body. So the only place we see this expressed, curiously, is the germ cells. So curious because the germ cells are the part, I guess, the part of the body that is responsible for the next generation. And one of the remarkable observations is the next generation is born at day zero. They don't inherit aging from their parents. So clearly, there's that compartment has a rejuvenative capacity, right? Or it has an ability to sort of reset biology in a way the rest of the body doesn't. So, I'm joining the dots in a way that I guess it may or may not be correct, but it's certainly curious that the only part of the body this gene is expressed is the part of the body that has to have a sort of rejuvenative capacity.
And it's called SB000.
Yeah, we called it SP000, which is a bit of a clumsy name, but it's because after the discovery of this gene, we basically concluded there must be other single genes sufficient for rejuvenation out there because this was just the first one that cropped up. And if it's that easy, maybe we should look at single genes systematically the way we originally intended to do. And we ended up finding 150 rejuvenation genes, so individually sufficient to rejuvenate a cell, according to our own biomarker. And we're testing 50 of those genes now in DNA methylation clocks in the wet lab, and so there's this 50 that came after this original gene. So it's a bit clumsy why it's called SP000, but I guess it was like the big bang of single genes, and then 1 to 50 is finding other things on the back of that.
Yeah, I mean so it feels like we're sitting talking about this like it's no big deal, but I mean this is could be hugely important. I mean, if you just zoom out a bit and look at the aging biology field as a whole, I mean the most promising thing right now, I think most people would agree on is partial reprogramming. And you're just saying that you found a single gene that can do that safer than the state of the art.
Yeah, that I mean, what's the biggest? What are some of the biggest question marks in developing drugs to rejuvenate? Well, one of the biggest is what's rejuvenation? Like we don't even have that story. We don't have that chapter open. So, what we have now is new chapters of aging and rejuvenation biology. And the moment that happens, right, we've just got so much greater traction on the problem than these mysterious transcription factors called Yamanaka factors that are really dangerous. So, once these new chapters open, there's a huge amount of things we can do and capability we have. So, I'm just going to caveat that that the reason I'm excited about having 50 rejuvenation genes is that inevitably these genes will fall down in different ways on the journey to developing a drug. So we might find that many of these genes have toxicity, like they rejuvenate, but they're toxic, like Yamanaka factors, that's a type of toxicity. Um, they may be hard to target with a drug. There's so many reasons that you're unable to develop a drug for a specific target, like P53. Yeah, this amazing gene, but we still don't have a P53 drug because it's just so challenging. So you need like a critical mass of targets just to survive the gauntlet of drug development and get something that people can take and actually benefit from these type of approaches. So I'm almost certain very few of those genes will survive the journey, but the fact we've got a critical mass gives me huge confidence and excitement that we will actually make the journey in the first place.
Yeah. And what about causality here? I mean, you can measure the biomarkers here in your single cell clock. But what can we? I mean, I guess we'll have to wait and see, right?
Yeah, so the This is a very valid critique, which is when you're using biomarkers to qualify whether you rejuvenated, it's incredibly easy to game your system. So you can end up basically reversing your biomarker, but that's all you do, right? You do that, it's like turning the hands back on your clock. You haven't reversed time, right? All you've done is you just gamed your system for telling time. So there's different ways we can get out of this situation. One is to test methylation clocks. So our single cell clock is based on the transcriptome. So now let's take a clock that's a different molecular layer, which is the methylome, not the transcriptome. You can then go one step further, which is do multiple methylation clocks. So does it work across methylation clocks? Obviously, you want to show the cell behaves younger. It's not just a clock that is younger. The cell's actually behaving younger. Um, and then, there's various ways to gain confidence. So we're just now in a process of gaining confidence via running basically orthogonal assays where each assay is as distant from each other as possible. So we gain maximum confidence that we're actually rejuvenating. I mean, I think the short way of putting this is if you've got loads of assays that all say that you've rejuvenated, um, it's like voting, right? It's more likely, the more likely situation is that you have in fact rejuvenated a cell. So we're just trying to do as many of these as possible.
Well, it could be a historic moment.
Yeah.
It might not be. Yeah, being a good scientist, it's very easy for me to get excited about this could be it, but there's also many ways these things can fall down. So we should reserve a bit of excitement for subsequent validation steps. But I think the fact this scenario even exists is sufficient cause for excitement because it didn't exist before, right? So it is a step forward.
This episode of Levity is brought to you by Vitalism Foundation. It's a community of action driven by Vitalism, a new moral philosophy whose first tenet is that life and health are good, death is humanity's core problem, and aging its primary agent. They're one of the few groups that truly understand aging as the emergency that it is.
Vitalism's goal is nothing short of monumental: to allocate 1% of global GDP to this project. It's ambitious, but it's exactly what's needed to tackle aging head-on.
As a member of Vitalism, you'll get access to a private community, exclusive content, and invites to special conferences. Plus, you'll receive insider prices on longevity wellness services from Vitalism's mission-aligned partners.
But more than that, you will be supporting a mission that's too important to leave to others to tackle.
You should really join us. Follow the link in the description below. For a limited time, you get 30% off forever. Just use the promo code LEVITY at checkout.
Again, that's 30% off forever with the promo code LEVITY. Let's make a difference together.
And you said to me when we spoke the last time that you think that scientific discovery up until now has been like serendipitous, but with the kind of technology you and others are developing, it might not be up to chance anymore.
Yeah, so there's two sides to this. So first is the enormous increase in throughput that a virtual cell or a cell simulation gives you. So you don't need to rely on serendipity because you can just do every experiment. You don't just have to pick and choose experiments, which sort of limits your chances. You can do all experiments. The second thing is every time we do a wet lab experiment and the simulation has predicted something wrong, that's the perfect data to create a better simulation. So all of that error is actually the perfect training material to make the better experimental system. So you end up in this positive feedback loop where no matter the outcome of your experiment, you're getting better and better at finding the answer that you're looking for. And that hasn't, this has happened in a very narrow way with scientists. You become a better scientist over time. You sort of learn what works and what doesn't work. But you can basically take the entire sum total of what you've measured and feed that back into these simulations and make them better. And so I think it's called lab in the loop or active machine learning, where you just, it's like the Terminator that just gets stronger as you try. So I think I've used the term Terminator to describe these things. I mean, hopefully that's not what it is in the long run. We're trying to make us live longer. Yeah, it's like the anti-Terminator. But you have this throughput benefit and you have this huge ability to learn from every experiment that we haven't previously had.
Mm. What kind of company are you? Are you an AI company? Are you soon to be a pharma startup?
Yeah, so very important question. We're the company that does whatever it takes so that at some point in the future, you wake up and your new reality is that each day you're actually getting better. So you're feeling better. One definition of that is I feel younger, right? It's sort of if you're getting fitter or you're losing weight, right? It's that similar trajectory where you're sort of feeling better, but actually getting younger, that's your experience, which is the opposite experience at the moment. Like all of us, we've all been younger and we have different ages right now, but we've been younger and we look back at that as a positive, right? Like, wasn't it great to be younger than I was today? So I'd imagine you wake up in the future and you're on a trajectory towards being younger. So whatever it takes to make that a reality, that's the company will be, right? To make that happen as soon as possible, which means sometimes doing the slower thing, as in, maybe we need more safety and that's going to take longer and we don't want to take a risk in a trial. We really want to get to the finishing line. So sometimes the shortcut is the long way. You just got to put in the work. So whatever we need to be in that new scenario in the future, that's the company we're going to be. So along the way, we've become more expert in aging clocks, and then later on more expert in these virtual cells. Now we're going to need to be more expert in drug development because we're now looking at lots of different options for what the drug's going to be.
Well, what's the time scale here? When are you trying this on people?
Very good question, and I'm almost certainly going to get reprimanded for this answer in the future. So I might as well just lean into it and accept my fate. Um, so it depends. It depends on the nature of the biology we use for rejuvenation. So this gene we've just found, it's a single gene when overexpressed, can rejuvenate multiple cell types according to methylation clocks at this stage. So let's just assume it is in fact real rejuvenation. Um, if we want to move from the experimental system into real humans like you and me, we need to try and do the same thing across all of our cells if we're going to actually substantially affect our bodies, right? We can't just do a tiny part of the body. We need to target the source of aging, which is each and every cell. We don't want to leave the heart behind or leave the brain behind. So if we're going to do that, we somehow need to get this gene to the whole body and overexpress it. So we now rely upon um, gene therapy vectors like AAV and LNP, and that's a whole world of pain at the moment trying to develop. It's like trying to build a road to all of the cells in the body. That those roads are being built right now, but they're not complete. And even if they're complete, they might not have become sufficiently de-risked from an investor's perspective, such that they will give you money to develop a drug using that road, right? So it sort of needs to be a road where there hasn't been any casualties, like somebody's gone along it a few times. So if we're overexpressing a gene, it's quite a challenging path to translating that into a therapeutic for you and me that does the things we're getting really excited about, like whole body rejuvenation. If we can find genes that we can inhibit that are rejuvenative, that is substantially better because it's far easier to throw a spanner in the works of biology than it is to deliver new biology to the whole body. So inhibitors are the typical type of drug used in pharmaceuticals, right? Inhibition is, you get these small molecule inhibitors, you get antibody inhibitors. So if we can find a way to inhibit biology that allows rejuvenation, that would be, I think some people have described this as like 100 times better. It would be like a 100x on this future I described. It's in terms of our velocity getting there. So after we found this single gene, we systematically went through single genes to try and find more rejuvenation genes. We actually found 40 genes that are individually sufficient to age the cell. And originally, we weren't interested in these, but um, from a drug development perspective, these genes are incredibly exciting because they're individually sufficient to age the cell. What if we inhibit them? Maybe we can rejuvenate the cell, right? We go in the opposite direction by inhibiting them and then rejuvenating the cell. But if we want to be able to do that across the body, we want to find genes that are expressed across the whole body. And only 3 out of those 40 genes are broadly expressed across the body, which gives you quite a narrow path. Um, but the really exciting thing is we look at those 3 genes, and the most pro-aging gene, when inhibited by known small molecules, increases lifespan in mice and reverses fibrosis in mice. So not only do you get this external validation from a sort of an outside study, but you see there's disease biology tied up with this, which gives you a clinical path, right, to developing the drug. Like there's a disease actually linking into this biology. Um, so we quite recently did our own internal studies where we basically validate, yeah, in fact, knocking down this gene with these small molecules or siRNA, which is a cleaner experiment, does impact fibrosis. And currently we have experiments underway where we're asking the question, does it rejuvenate the cell? I can't give you the answer, but all I can say is it's very promising. And again, I'm just going to say this, you know, this expression, how easy could it be? Um, you've got to entertain the idea because it could be a lot closer than you expect. So, the dream properties for a therapeutic would be small molecule that goes everywhere, that's an inhibitor that affects a disease and is rejuvenative, and obviously is safe in the process. And it looks like a lot of those boxes could be ticked already, and we've got relatively few boxes left to tick. Again, lots of things can fall down through drug development, so we almost want like 50 versions of that to make sure we get all the way to the finishing line. But yeah,
I mean, there are other companies doing fantastic work when it comes to drug development and using AI. I think in silico, for example. So, and I guess they have a whole platform that they offer other customers as well. So, I guess you have a huge advantage there in order to try to pin down what the genes that are most efficient and least toxic and so on, compared to how it was 10 years ago.
yeah. So once you know the targets, once you have these new chapters of aging biology, like those 40 genes, and rejuvenation biology, these 150 genes, there's a huge amount of built infrastructure in pharmaceuticals, like in silico medicine is one example, that you can now bring to bear on your specific problem. I'd actually say that infrastructure gets far more leverage on these targets than, say, traditional drug targets. But this is the great engineering project that follows the great science project. Like once you know what the biology is, because you've cracked that problem, the great engineering project takes over. So we're in this transition phase between, look, we have these new chapters of biology that can now be leveraged with this amazing sort of drug development infrastructure that's out there. So yeah, it's like it's the beginning of something. It's the end of something, but it's the beginning of something that's, I would say, more exciting, which is making it real.
When you hear like 140 genes plus 50, of course, you're not going to develop drugs for all of these targets. But do you foresee that will take like a handful of rejuvenative pills in the future, or how do you see that?
Yeah, so we're gonna gear up for both scenarios where, it how easy could it be? So let's just assume it's going to work, right? Keep pushing that forwards until it falls down. But also, look, this stuff looks great now, but it's probably not going to work out. So let's make sure we've got some backup plans. So let's keep trying to find new biology. Maybe 190 is half of what's left to be found. Like there might be more biology. So just keep everything going until we get the first thing across the finishing line. Because we don't know, right? We can't qualify what's good or not good until we're very close to this being real for humans, like maybe in a phase 1 trial where people are taking it and measuring their clocks and measuring other markers. Like we can't really qualify what's good and not good from this very early stage. So we've got to just keep the whole mill going, and you can keep training, you keep running the loop, right? It's like everything keeps like iterating and getting better. Your tools are getting better. So you keep that machine going until you start getting that early success, and then hopefully you have a second generation, a third generation. Um, yeah.
I'm not sure I understood completely. So you said you can identify a cell, a gene that if you overexpress it, it can work as a Yamanaka-factor, but it rejuvenates the cell. But then you also talk about you can inhibit certain expressions. But then is that also rejuvenation then, or are we talking about different things here?
Yeah, so we are talking, the definition of rejuvenation is the same, which is, does a single cell clock or does a methylation clock go backwards? So it's the same definition, but the mechanism is different. So Yamanaka factors, you have to put more Yamanaka factors into a cell, right? That's you basically overexpress those genes and you get rejuvenation. Inhibition, some genes are expressed and you're interfering with their functions, right? You're basically, it's like a spanner in the works. You're stopping them doing what they need to do. Um, and that's just easier from a drug development perspective. It's easier to develop drugs that basically disrupt what proteins are doing rather than trying to put new proteins into a cell. But the definition of rejuvenation is the same, right? Does the cell get younger or older according to single cell clocks and methylation clocks in our case?
But inhibition doesn't mean that just means that they age slower. It also means reversal, and older cells become younger.
Yeah, well, so that's the experiment at the moment. Does it mean reversal? Can we inhibit a protein?
Yeah,
Like, does this, just because we overexpress this gene and it ages the cell doesn't necessarily mean if we bring it down, it rejuvenates the cell. It might just
yeah.
stop whatever that process is that's aging the cell. But maybe it is bidirectional. So maybe here's this set level of protein or function of this protein. And if you put it up, there's more aging. And if you put it down, you actually go the opposite direction. And again, I can't tell you the answer, but um, it's looking promising.
All right, yeah. So inhibition as a tool of rejuvenation. That's the question.
Realm of the possible at the moment. Yep.
I was also thinking rhetorically, if you talked about the funding and we want a whole body to be rejuvenated, but I mean, if you could rejuvenate the skin cells, then that would be a proof of concept that would attract unlimited amount of investments, I'm sure. Plus,
No,
we're very good
absolutely.
at identify age in skin. I mean, we can just see it. It's something that everyone would understand immediately.
Yes,
Sorry.
this is a very good point, Patrick, because this sort of comes back to the question, what's the type of company you want to be? So you have to look very closely in the darkest depths of your soul. Like, what is it I'm going to struggle for 10 years? Because that's what it's going to take to deliver a real product, right? You've got to sort of gear up for that. What is the thing that I'm uniquely excited about that's going to be worth the struggle? Because it's got to be worth it. Otherwise, you're going to give up halfway. So the thing that's worth it for me is rejuvenation across the body that substantially increases lifespans. Obviously, the healthy version of that. I don't want people to be trapped in Alzheimer's or trapped in, a poor state of health. That's what gets me excited. That's something worth struggling for. If along the way somebody wants to specialize and do the skin, that's great. They can come to us and we can help them do that. But that's not what we're going to do internally as a company. There's sort of it's a very specific line, what we do internally, as in what we're excited about and what we're willing to struggle about, which is basically the whole body at once, or as much of the body as we can pick up. Anything that isn't rejuvenative or that is a narrow part of the body is something that we would look to others to help us with.
You can still imagine that you have to decide on if a pharma company is going to develop a drug, they need to decide on some sort of indication, right? How do you think about that?
Yeah, so we're very fortunate that the biology took us to an indication. So, there's two ways to choose an indication. One is to look at like 15 variables at once across all the indications, because there's lots of age-driven diseases, and try and like be a Rain Man, yeah, and then like choose the best one. And that's incredibly complex. And there's whole consultancies that will charge you like six figures to give you the wrong answer because it's a moving target. So like I guess we struggled with that for a long time, and then almost it's almost comedic. Just by doing a systematic screen for the biology and finding the new chapters of aging biology and rejuvenation biology, it took us to the answer. We just needed to know what the biology was, and this biology, and we're not the only people that have noticed that the biology of fibrosis is tied to the biology of aging. There was a study last year about interleukin 11 or IL-11, so it's this circulating protein. Originally, researchers working on fibrosis noticed that samples from patients at older ages had higher levels of IL-11. So these patients all had fibrosis, but more IL-11 in the older patients. So speculatively, they got rid of IL-11 genetically or by antibody, and the mice lived 25% longer. And it's called the super granny mice.
Yeah.
So there's precedent for fibrosis biology and aging biology to sort of coalesce, and we sort of see this in our screens as well. Um, but that's like this dream scenario where the biology we want to work on, which is aging biology, is also offering a development path within today's world, which is working on diseases one at a time, not preventing diseases, which is harder to monetize. So the data gave us the answer, not us trying to second guess what the answer was.
I mean, I love it that you have this passion, really, to solve this problem, and you have had it for a long time. But I guess that passion is. Can you take that passion to investors, and when you seek funding?
So far, I have been able to, but like at every new step of fundraising that's required, it's a different audience. They have different requirements, so it remains to be seen whether I can convince people that are increasingly different from me. Yeah, my original investor was a postdoc scientist originally, right? That sort of he became an entrepreneur and he ended up running a public company, and like he sort of had to like a caterpillar becoming a butterfly, like go through this metamorphosis as an individual to talk to people properly at these different levels. So um, I think communication is now one of the points of maximum leverage. Like, can I translate my enthusiasm into like you plant a seed in somebody else's head who didn't previously know this was real, and isn't familiar with the language. So, um, I mean, one thing I can do, one thing that helps this is just making making what we've done more real. So show this works in a mouse visibly, right? Not just with clocks, visibly. Go into a larger organism and show these mice behave younger. Like you can actually just, if you hit a new watermark in terms of what you've achieved, that just speaks to more people. You don't have to, you know, you don't have to twist yourself in knots trying to get across to people where there's like a divide that can't be bridged. So that's what we're trying to do. Take these things we found and as quickly as possible, see could we get this to work in a very, in a way that speaks to way more people that are going to need to help us.
In just a few years' time, there's been a lot of developments in artificial intelligence in general. I mean, we now have these reasoning models. They're happening a lot with diffusion models. Um, how do you keep track on that, and is it getting increasingly useful for your line of work?
Yeah, so this is a really exciting frontier of science, and I'd argue we're sort of we're trying to make the most of it already with these virtual cells. It's not AGI, it's narrow AI. It's basically taking the amazing pattern recognition of these models and applying that to a cell and allowing us to basically do things inside the computer. So it's sort of narrow AI. Um, I think there's a lot going on with the power of these models, and are they the agentic side of it? Like, are they able to reason better than human scientists? But I think the best way to put this is that ultimately, any powerful intelligence needs the data to make a good decision. So, like, yeah, if you just took the data that was out there at the moment, it's just Yamanaka factors. If you gave an infinitely intelligent agent that data, it would still not be able to go beyond the Yamanaka factors. You need the tools to ask the question, like, what is a rejuvenation gene? So you need big data sets to achieve that. So, um, I mean, ultimately, it lies in the generation of big data sets, which allow you to create new tools. And I think if the AGI arrives in 2 years and makes all of the next 2 years of work futile, like effectively it does the same work that we're going to do in the next 2 years in 10 minutes, um, it will still need the data set. It will still need the data set. So that will be our last stand, right? We'll be negotiating with the AGI to eke out another year of life on the back of the data set we have. That, I mean, I mean, I, that's an increasingly mainstream sort of position, at least in the Bay Area. I'm not all the way there yet. I think that I think our sort of mushy biological brains have got a lot left to give. But ultimately, you need these data sets, and that's where most of the investment of the company goes. It's generating data sets which create single cell aging clocks, which create better virtual cells, and then running high throughput experiments, feeding the data back. So those data sets, you can't fast forward them. They take a lot of capital, and an AGI will need them to do a better job than we're doing.
I guess the AGI could create a humanoid robot and create lots of humanoid robots to do the lab work for them.
Yeah, yeah. I think it's coming, but we also need to be prepared for a scenario where that happens slower. So I think there's a lot of people create this silver bullet where it's sort of they end up taking away their drive to work on the problem. I don't think that's helpful. I think we should always assume it's going to be very hard and prepare for that. And if it's easy, that's great. And we should also experiment with these new tools. But we should always be preparing for both scenarios.
So about the study, I guess it's a curious situation now when we're recording this before you've released it. But two things: is there anything about the study? Of course, we talked a lot about this single gene that you found, and but is there anything else in the study that we haven't discussed that you feel worth mentioning? And also, what are your hopes now that you're going to publish it?
So I'm incredibly embarrassed to say I haven't even been able to read the paper yet. And part of it's negligence, but also I'm aware of the findings. It's just they're presented in a much better way, right? Like I've looked at the figures and it's like, wow, this is like a real paper. Like I'm very impressed by my team, like the amount of efforts that's gone into it. So I can't really speak to the darkest depths of the paper at this stage, apart from there's plenty of work to do. I think we just want to be clear about this. Like, we have these findings and there's other things we want to do. So for instance, if this is real rejuvenation, and my definition of that is you reverse age by 5 years, and once you release the treatment, it takes 5 years to get back to the starting age. Yeah, so like that's the most comprehensive definition of rejuvenation. Like, it's not just you release the treatment and you go back to your starting age like that quickly. So if it's real rejuvenation, we need to do those types of experiments where we express the gene, we release the gene for say 4 weeks, and then we see how much rejuvenation is locked in and how much was very fragile and is basically dependent on continuous exposure to the drug. So, we want to explore that dimension. We want to go into mice, like make sure this works in living organisms. We want to see, these genes where this will be a later manuscript perhaps. Like if we can inhibit genes, is it rejuvenative? But also genes that are expressed across the body, which is very helpful if you want to inhibit and rejuvenate. Those types of genes tend to be essential. They're expressed everywhere. They're probably important, and if you mess
Yeah.
around with them, that might be dangerous. So again, like this is another reason it might fall down. So there's all these ways. There's all these remaining question marks that we're now busy on. So I think it's just important to caveat what we found with what we haven't found yet and what we
Yeah.
haven't proven. But I think it's cause for excitement.
If Jeff Bezos is watching this program, what would you ask from him? Do you have everything you want, or what do you want to carry out your program? Ideally, if there were no limits, almost like what would you ask for?
I think I think the way that we're structured as a company is we achieve a milestone, we go to the markets, with a higher value, ideally, right, on the back of the milestone, and then we raise some more money, and we repeat, right? We keep going like that. Financing is incredibly time consuming. Yeah, it does take you away. You just have to use a lot of your brain for that, and you might take your eye off the ball of something else. Um, you can manage both, and there's people that effectively manage both, but it is very time consuming. So that's the advantage of having a huge pot of capital up front, like an Altos Labs, right? You can just steam through all of the steps without changing gears. It's like an automatic transmission, right? You just keep going to the next step. I'm not a silver lining person, but the silver lining of having to answer to people through fundraising is that forces you to focus, forces you to prioritize things, forces you to remove dysfunction or bad projects. Um, and that seems to have stood us in good stead. We're actually making a lot of progress here. I think we've got sufficient capital. I don't think we're undercapitalized, but we do have to go back, and you never know whether you're going back into a storm. Like you might not be able to get it as easily, or you just don't know what's happening. So it's like a financing risk, which you could remove, right, if you had someone without, well, effectively unlimited resource. And then I think it would just be great for people to know what's going on. So when we achieve these milestones, if people understand the significance, the real finishing line, which is waking up one day and these drugs are real and we're getting better, um, we just minimize the friction. Yeah, like we just, if everybody knows the significance, they're helping you get there. They're not, maybe fighting against you or being skeptic. And I think that's the most important thing. People understand what's going on, and that's part of you know part of why these podcasts are important, right? Like getting the message out there, and then some people translating what I'm saying into a better version of what I'm saying to a different audience.
But I mean, you could not use a cadre of programmers or lab workers. I mean, you don't need more people or anything like that, or
Um, so yeah, I think more people is more. More sometimes is more, but sometimes there's too many chefs in the kitchen, and you need a direction, you need leadership, and small teams can be very good, can be the optimum solution to certain things, especially in science, especially in very sort of deepest, darkest depths of science where things are very challenging. So yeah, I think maybe more people, but I wouldn't say that's the bottleneck. I say right now it's just the bottleneck is just increasing confidence, which is basically scientific experiments. Like, how do we increase confidence? That basically creates the scientific experiment is the best answer to doing that, right? Like that's where experiments come from. It's like, well, we can't get any more from the literature. The best thing to do is get in the wet lab and ask our specific question head on with the best tools available to doing that. So it's just systematically answering these questions, and then it becomes engineering. So I think one thing which would be really amazing would be the whole infrastructure of the pharmaceutical world plugging into what is now this new biology and giving it the best that they've got to make this drug of the future, right? Like if they felt the way I do about making this a reality, I think that would be probably the best thing that could happen right now, because they have all the tools for what we'll need in the future. Yeah, so if they bring that to bear on this problem, things will move much faster. Like they have the expertise, they're just not quite there in terms of like feeling the excitement and therefore acting on that excitement.
in terms of laws and regulation and policy, are there any bottlenecks there that you'd like to address?
Yeah, so I want to draw an analogy to GLP-1 agonists, so these anti-obesity drugs that I think everybody knows about them by now. So, if you look at the sort of back history of these drugs, once upon a time, there were doctors and they were receiving obese patients and they were having to treat secondary complications of obesity, and they didn't really have much they could offer the patient, right? And it was just a situation where like the primary issue was the obesity, but they couldn't address it. And then what happened was these drugs originally developed for diabetes started to show really interesting secondary effects like weight loss. And so you had a regulator that now all of a sudden they were in the middle and they had a win-win situation, which was you have clinicians with real clinical need trying to treat their patients but not able to, and you have existing technology developed for other disease but could have a real benefit to these patients. So at that point, obesity was reclassified as a disease and it was a win-win. So now you have all these patients, they're receiving drugs, they're getting, they're losing weight. That's good for all the secondary complications. The pharmaceutical companies obviously benefit as well. Um, you can easily imagine in the future, doctors are dealing with a inflow of aged individuals not able to treat the secondary complications of aging like Alzheimer's, Parkinson's. And on the other side, you have a drug developed for fibrosis, but aging or rejuvenation biology originally, yeah, but it developed for fibrosis. It's being used to treat fibrosis patients. The regulator's in the same position where they say, look, if we reclassify aging as a disease, these clinicians can now distribute this drug and we don't have all these expensive secondary complications. And the drug developer, whether that's Shift or somebody else, benefits and this becomes a new industry, right? GLP-1s is like now effectively a new industry. So you can imagine an analogous situation in the future. So of course, if people make this argument earlier, the regulators can be more primed for this happening and they can reclassify earlier. So I guess that would be helpful. But I think once you've got the technology there, you get out of this chicken and egg scenario where the regulators, well, like this is all a bit hypothetical, right? Reclassifying aging as a disease because where's the therapeutic, right? But if the therapeutic's there, then you put those two pieces together and all of a sudden you can distribute to everyone because everyone has aging. It's like, it's one of the unifiers.
So it's not a disease because we don't have a medicine for it. Interesting.
I would speculate the moment we have a medicine, the definition may change quite quickly to disease or indication, I think is the better way of putting it. It becomes an indication.
I was thinking about your name, Shift. Was that something you did you have that name from the beginning, or is it something that came later? I mean, it's really fitting now, your name, but it feels like it must have been for some other purpose if you had it all the way from the beginning.
Yeah, so the term shift is used to describe an incredibly esoteric phenomenon called mitochondrial DNA heteroplasmy shift, which I hope you forget. I hope you forget. So earlier on, I said that originally I thought that the cause of aging was damage to the mitochondrial genome, and if we got rid of that damage, we effectively solve aging. So the phenomenon of decreasing that damage is called mitochondrial DNA heteroplasmy shift. The reason being there's thousands of copies of mitochondrial genomes per cell. You basically have pathogenic genomes as one population and good genomes as another population. So you can basically shift between different proportions of those two populations. So that's where shift comes from. Along the way, it seems to be quite a versatile word that you could use it to describe a company that pivots a lot. We only pivoted once, yeah, but it's sort of that was appropriate. And then shift is sufficiently generic that we've never had to discontinue it. So I don't know whether there's some sort of poetry of the universe where it becomes the ideal word at the end of the journey, but it's certainly still relevant. So we've got no intention of changing it at the moment. Yeah.
And after this study is published, are you looking to do another fundraise now? You've raised $16 million so far, if I'm correct.
Yeah, so most of that $16 million is relatively recently. So after we started focusing on improved factors for rejuvenation, like improving beyond the Yamanaka factors, our decision just predated the launch of Altos labs. So all of a sudden, Altos basically legitimized this field as an investment opportunity. So that allowed, it allowed us to raise capital much more easily than we had previously. Sort of moving forwards, we are raising right now, but we're raising a surprisingly small amount, and the reason is we only need a lot of money once we want to develop a drug internally. So a drug that has these properties, small molecule, gets across the body, inhibits a gene, rejuvenates and affects a disease, and does that safely. Like that is the set of properties that we need to be sufficiently excited to struggle for 10 years and make a drug real. Yeah, that's what we need. That's that's what will be required to deploy capital. Yeah, one of those programs. Up until that point, we just want to assemble as many options as possible and choose the best starting point that that gives us those properties. So here's a target and here's a molecule that's used for that. So right now we're in relatively modest cash burn where we don't need. It's not. I'm not saying it's small amounts of capital because I think it's $15 million we want to raise to just get an extra year, which basically allows us to get all of our options together and basically, what's the best possible start point. So that's all it does. But relatively small amount. But the moment we find the drug of our dreams, that then becomes a much larger raise. But the way that I see it is if we hit these milestones, essentially you can raise the capital you need, right? If you actually move the front line, the money will be there. I know sometimes it's more challenging, sometimes it's less challenging, but ultimately, if you move the real front line, I think capital is a secondary problem at that point. The capital will find you. There's plenty of capital out there. Yeah, there's a record amount of capital out there. It's just trying to find a home, and the home is where the progress is being made. Obviously, you've got to communicate that progress as well, which is, that's one of my challenges and the big bottleneck I'm trying to work on. But but that's when we would raise a much larger sum of money. So we're just looking basically for a small amount of money. Maybe someone wants to join before the whole thing takes off, right? So we're sort of looking for people that are ideally more mission oriented than me, right? They're more excited about the finishing line because it's good to get that mission embedded, right? Sort of more broadly. So yeah, anyone that's out there that's excited about this, there is like this last opportunity to get on board before things start moving relatively quickly. Of course, we might encounter issues, but that is the risk of these types of enterprises. But it doesn't mean it's not worth doing.
I mean, you're completely aligned with what we are doing with Levity. We don't want incremental steps. We don't want some health span benefit late in life and then still die from an age-related disease. We want real rejuvenation. But I also feel like there's a lot of people in this field that are not ready to use that terminology. They're afraid that they won't get funding. They're afraid that they won't get taken seriously, and so on. But still, here you are, Daniel, and doing this, and you're very. You speak in the open about how you feel about this. So why isn't everyone doing this?
Yeah, so you're not the only one that's picked up on this. So there's a lot of people that are closeted about what they're excited about. So it's like a catfish scenario, right? You show up to substantially increase lifespan, and then you get comfortable with what sounds a lot like medicine today. Yeah, like we just want to keep people healthier for a little bit longer. Um, but sort of you have to be quite honest with yourself, like what am I really excited about? Because otherwise you're gonna tear yourself to pieces later. You're gonna be struggling for something you don't even want. And I think if we want to reach the finishing line, I guess we just need a bit of courage in the short term because it's much faster to just, you know, be authentic about it and find authentic people to help you, even though you might not get the most immediate interest or most capital in. The world does need to catch up a little bit. That will always be the case with something new, especially in this type of context. But that slower movement is still faster towards what we really want, the exciting thing. So I think it's Mark Hammerlayan says there should be a longevity pride day where everyone that's because it's sort of very similar, right? Where everyone that's closeted about this just is proud about it, and then everyone that sees that and they come out, and then it becomes I'd say this is becoming more mainstream now, but there's still a lot of people that are very closeted, and I don't think that minimizing risk by saying nothing about what you're excited about is the correct answer. I think you need to manage the risk. I don't want to say crazy stuff like I don't know. I mean, I don't know, live forever, because then the default headline is Daniel helping billionaires live forever.
Right.
Daniel helping, that's the clickbait title. So there's better ways to frame it. Like, would you like to wake up every day and feel better on an ongoing basis, right? Like that'll be the common experience, right? Like if we actually make, so yeah, there's better ways to frame it so we can make our life easier whilst trying to bring people on board. But I think imagine the technology exists. Imagine like today it exists. It's almost mundane. There's a drug, and you could basically reduce your risk of getting very horrible diseases, which are like now historical artifacts, right? These things that you just read about. You can now go and do that, or you can be reckless and you could just not take it, and maybe you just want to annoy a family member. I don't know, whatever your reason, right? But if these exist and it's basically become mainstream, that I think that would be the obvious thing to do. Um, and then there's an interesting set of problems that arises in that situation. Like, if you are to live longer, are you able to uh, carry all that baggage of life into the next day and be a nice person, or are you just very grumpy person and you can never treat somebody in good faith because you've just been betrayed all that, those past 300 years, right? You've been betrayed in every different way, and you're never going to treat the next person like that. Anyway, there's lots of interesting problems, but I feel like the secondary problems are much better than we're all going to die and probably die a horrible death. And even if we don't get a horrible disease, we're going to be wearing an adult diaper, which is just such a tragedy. And it doesn't need to be that way.
There's also another story here. The other story in the longevity field among those who are aligned with us is that it's taking too long, and there's an increasing interest among aging biologists in cryonics, for example. What's your timeline here? I mean, what you're talking about sounds a lot like longevity escape velocity. You wake up and you're younger. When should people start to get their hopes up about that?
Yeah, so forgive me, Peter. Patrick asked that question, and I just forgot to answer it. I didn't dodge it. So, um, if we have to overexpress a gene, I think we rely upon the whole ecosystem of gene delivery, gene vectors, and I would say we're like 15 years away from getting this across the body. If we can inhibit a broadly expressed gene with a small molecule, I think that's a fast forward of 10 years. So you could, if you find a molecule with those properties, you could have a phase one trial, which is basically healthy people. Remember that, right? In phase one trials, you give a drug to healthy people because they're better able to stand side effects. So you have healthy people taking a small molecule drug that can rejuvenate most of their body, and they'll be measuring their methylation clocks. And thanks to Brian Johnson, primarily, these clocks are now a mainstream topic, right? Like that, he's the champion of the clocks at the moment.
Yeah.
Um, so, thanks to him, the readouts in this trial will mean something in a very sort of mainstream way, and that's the plot of a science fiction film called The Last Generation to Die. It's the first clinical trial, and people are taking these drugs, and one of the patients is the dad of the principal trial coordinator. She gives him 10 times the dose. He starts rejuvenating. Uh, he gets into a fight. Even though he loses the fight, he can't believe how well he did. He's staring at his fists, and at that moment, he realizes it's worked. And that's that's the specter of that phase one trial. People are taking it, and you're reading this headline, and somebody's reporting their clocks have gone back beyond lifestyle
Yeah,
and
it's so
diet.
interesting because, yeah, as you said, phase one is like the safety trial, the first safety trial, but here you would show immediate efficacy as well, right? If it works.
Yeah, it wouldn't be the primary endpoint of the trial.
Yeah, of
That would
course.
be safety, but of course you would monitor these things,
Yeah.
and that would give you a data set that you could then keep, you keep building that to make the case to the regulator that we could do some real good much sooner, right? Because this is actually doing two things. It's a disease, potentially fibrosis, but it's also something that's beyond the typical realm of medicine. Um, but you just imagine that, that trial's happening, and it's a very special moment, and that's why it's this. I think it's yeah, it's Last Generation to Die. It's an amazing script. I just wish. I think they just need the funding to make the film, but it's just an amazing plot, and obviously I'd be in the audience watching that 10 times, like just to make it real. But then just last point on this, which is there is a selfish aspect here, which is the reason I get excited is that once you make these things real, it's like a sonic wave just passes through the atmosphere, and everybody at that moment in time almost freezes at their age, or perhaps they reverse. But I would make the argument that it's increasingly difficult to reverse because if you lose cells, you can't resurrect them, right? And that's where replacement comes in. So the sooner this technology impacts your own body, the sooner you're going to stop irreparable processes or irreversible processes. So I was I used to be younger. I'm not old, but I used to be younger, and I preferred being I preferred being younger. So I want this stuff to interface me as soon as possible. So now we're looking at properties that look very much like the drug of my dreams. And again, it can fall down in multiple ways. You just feel that you feel that force, you feel that sort of magnetism, like this is real, and therefore I'm want to work harder, and it's this positive feedback loop, right? The closer you get to it, the faster you go, and then it becomes almost certain, right? And I think we're getting there, but with these steps forward in the biology and then the drug development beyond that, you're going to get this positive feedback loop where it becomes inevitable. I'm not saying we're there yet, but I think we're getting there, and we're not that far away.
Now, you haven't read your own study yet, but I can tell you that you mentioned the fountain of youth in it.
I do know that we did deliberately cut down the flowery language and the colourful language because we want to speak to the people that are closeted, but we also want to speak to people that are on the fence and might be turned off. So that there's a way. We did include it because wouldn't it be strange if you didn't use the superlative that everybody it's the common reference point, right? Like that
Yeah.
means something to a lot of people, so it's a tool, and we shouldn't be shy about using the tools. Maybe there's a risk to doing so, but um, so be it. So be it. As long as we're moving to the real finishing line faster, so be it.
Emil Kensiora, who's the CEO of Tomorrow Bio, he's of course a little more skeptic about the timeframes.
Yep.
What do you think about cryonics? Is that going to be needed for people, say, that are like me and Patrick in their 40s and 50s?
I think Aubrey de Grey touched upon this point very well at the Vitalist Bay conference recently. So, when we talk about these technologies, we're actually talking about lots of people at lots of different ages. So you could argue if somebody's 2 years away from, you know, the lifespan of their parents, yeah, like they cryonics is a much better solution because it's better than being buried in the ground, and it's better than being cremated, right? Um, if you're a long, long way away from cryonics, I guess there's less you just feel less of a need to choose that option. Like you might want to just bide your time and see what happens in your remaining lifespan. So I think there's a different answer depending on where you are on the spectrum. I think ultimately, until we've got all solutions working, we can't second guess which the best one is.
Yeah.
So I think it's Jean Hebert. I sort of had a discussion a long time, like the pitfalls of reprogramming and how replacement is a great answer. And I agree. I think both are answers. They're different answers, and we should pursue both and not second guess what the best is, because otherwise we end up being very fickle and flip-flopping, and we don't nourish either option sufficiently, and then both die, and we don't want that. So I think we should always have diversity in approach, because otherwise we get in a very fragile situation where it turns out that doesn't work and we don't have any backup plan. So I think all of these approaches are good. I know that's an incredibly unspicy take on it, but I think they're all good, and they're cryonics much better for older people. I think that would be my controversial take on it.
I'm going to try a pun here. Are you going to do you think that you're going to shift the whole field now to go away from Yamanaka factors?
I would never use that language, because I wouldn't be able to live with myself just watching that. I'd probably explode. I'd probably explode from the cringe wave. Anyone is free to use that language. It's sort of funny. A newspaper headline will, like, they live off those types of things, right? Like, they're just trying to make it good.
In my previous life, I used to work for a tabloid.
I also, people use it against us. They said, "Oh, they're like Shifty Bioscience. Why didn't you talk about this thing? You slippery character. You're so shifty." I quite frequently, they miss out the F when they're talking about us, and
Oh,
and that's funny, and that happens. There's other things, like when we started getting involved in AI and machine learning. Um, we thought a great idea would be to rebrand as Deep Shift, but removing, and then we could be Cat Shift, and we could be Dog Shift, could
of
be,
course.
for the, we're going to target those animals. It could be Horse Shift, and we didn't even realize, because the investors in the room were like, they're just completely silent, and we didn't even get it right. That's sort of speaks to our personalities. But I'd love to launch a sub brand called Dog Shift.
Yeah, sure. I mean, dog rejuvenation is a thing now. So
Exactly, but at this point in time, I think that will be used against us, and that would be discrediting, right? Because it'd be like, are these guys even serious? Is this just a stand-up? Is this a stand-up comedian pretending to be a startup entrepreneur? So I don't want to. I mean, I know that's a niche that needs to be filled, but I don't want to fill that niche right now.
I did have a serious question there, though. I mean, you could imagine that if you create the state of the art here, other reprogramming companies would follow.
Yes, it's inevitable. Once there's a better way, if people are being rational about it, they will want to get to the interesting bit, which is what's next. So that's a constant risk as a startup, that you basically X marks the spot and you have fast followers. The way to deal with this is IP. So you find your genes and you make claims around those genes, so that if somebody does develop the drug, and they didn't put the work in, they basically just lent on your work, you can make a claim to that. But tend to be court cases, so nothing's for certain, but there are ways to protect the research and development costs, and mostly the time that's gone into it. I mean, what I would say is, join us, right? Partner with us. If you want to do the exciting bit, take what we've done, and I want to get to the finishing line as fast as faster than probably anybody else. I'm not going to create a toll road with a ridiculous fee, but we do need to pay our research and development and stay alive, right, until the glorious finishing line is reached. So come to us, and if you think this technology can be deployed in a way that we haven't thought of, like, talk to us about that, and we can see if it's a win-win situation.
Yeah, Patrick.
Yes, we've made history here, hopefully.
Yeah, of course. Daniel has put in a lot of caveats today, so I guess we can say that Daniel has made history.
I got to give credit to the whole team. So
Of course. Yeah.
my job now is to show up and take credit for everything. Yeah. Um, it's just the nature of delegation, right? I have to
Mm.
be the face, and then but ultimately, um, I started off with a hypothesis that was flawed. It was actually one of our interns, Brendan Swain, who got interested in single cell data, created a single cell aging clock. Lucas Camillo, who joined us more recently, created a human version of that, mastered cell simulations. Lots of wet lab scientists did really amazing things to actually test these things in the dish. There's so many stars behind the scenes, and they're all on the paper. You'll see sort of the contributions there, and then in subsequent publications there'll be sort of different things where others have been instrumental. But there's a big team effort. We're a relatively young team, so this is the first time we've a lot of these people, this is one of their first achievements. They're not resting on their laurels. This is like how they're proving their worth. So it's a lot of great people that you haven't heard about. So a lot of credit to Brendan, Lucas, I think Kate Maskalenka, and Rehab Garm, who are on the paper as well, right at the top. And there's many others, but they're all on the paper.
How many are you working on at Shift now?
It's 24 of us. A couple of us are part time. Um, I'm definitely not part time. So yeah, 24 of us. We were only 8 of us last June. So yeah, very big for us historically. At the beginning of Shift, it was just me, and family and friends helping me. So I think we're going to stay that way probably for the next year or so. But once this is drug development, things have to move very quickly, and you need the best talent pool for taking a drug from here to clinical trial. So I think the shape of the team starts changing very quickly at that point. But yeah, 24 of us at the moment, all in Cambridge. Well, most of us in Cambridge, 4 of us in Toronto. Basically, the machine learning team in Toronto, because our advisor Bo Wang is one of the authors of SCGPT, one of the cell simulations that we use. So we have a team basically that's co-located with Bo Wang. So we're getting the very best from the world of cell simulation.
He's doing really interesting stuff now.
Yeah,
I mean, he shows up in my social media feed all the time, and I feel like I should really do a deep dive soon on his work.
yeah. I mean, there's this whole new world of taking these amazing pattern recognition machines and plugging the biology in. Like, how do you plug biology into this pattern recognition? So, like, Bo's basically taking as much biology as possible, like visual images, let the machines look at that. Single cell data, let's let the images look at that. Machines look at that. So yeah, Bo Wang is basically creating the adapter between the fast moving machine learning world and biology, which is less fast moving. But he's the one building the bridge, and he's doing a great job of doing that. SCGPT is just one of many things he's done. We know him only for SCGPT, but he's quite well known beyond SCGPT. So yeah, he's one of the reasons why we're finding these genes.
Yeah.
Yeah.
Okay, Daniel. What we usually do at the end of our episodes is we ask our guests to recommend 3 books. Is that something you've thought of?
A little bit.
Yeah.
I'm not going to pretend. I have been negligent in preparation for this interview. I managed to get the headphones on and I managed to get a quiet space. Yeah, so good books. Good books. I think, I mean, I know this is increasingly controversial, but um, Elon Musk's biography, I think it's by
Walter Isaacson, right?
Walter Isaacson, that's right. Um, just sort of it gives you the anatomy, like how did these amazing things happen? So you sort of go upstream of that, like, all right, here's this individual, like, how's he doing this? What's his history? Um, and whether that's good or bad, and just sort of getting the whole anatomy of this like who's doing big things on behalf of others. Obviously, there's controversies, right? Like he has opinions, but you shouldn't deny we're very lucky to be living in a time where that guy exists and he's doing these things, and we're going to benefit from them. So I just find that individual absolutely fascinating, and there's some really troubling things he went through, right? That sort of perhaps reflects why he's able to do these things. And obviously you don't want to go through some of these horrendous things yourself, right? To reach your goal, like, but you want to sort of learn from it. So I think that's just understanding that individual is that book is really great. There's sort of just a lot of analogies to what we're trying to do, right? Like going all in at every stage, like not just say that I reached a an oasis of peace briefly, right? I could just sit there and enjoy myself, or I could take my new position and go all in to get to where we really want to go, right? So he's just got, the amount of courage that I mean, maybe it's recklessness, right? Depends on who you ask, but he goes like all in each step, and I think that's a very inspiring sort of template if you want to achieve very hard things. Like, we're in that category of trying to achieve very hard things. So that's one of them. Um, yeah, I'm negligent, I'm afraid, so I don't have any other ones that come to mind.
That's okay, Daniel. I was thinking about Elon. Yeah, he's not too fond of our interest, though. He kind of feels like we should die to make room for the next generation. So we still got some convincing to do there.
Yeah, I think I'm, I it's I don't want to. So there's lots of individuals that hold strong opinions, but there's a subset of individuals that hold strong opinions but are incredibly open to uh, reasonable arguments. So, you could make an argument, and that person who's rational will, quickly admit they weren't thinking about that, and they didn't think about, this bigger picture, and they will change their opinion, and they will hold that as a new opinion. I like to think Elon's in that category. He has very strong opinions, but he hasn't seen a sufficiently strong case to entertain this because, I think if I was going to try and explain this on his behalf, because you should really ask him, right? It's not for me to speak on his behalf, but um, this field is sort of an attack. If you are a high net worth individual, it's an attack vector because you can frame this as a selfish pursuit, right? You're just trying to live longer. Like, you don't want the party of life to stop. You've become a billionaire. It's great. Why would you want that to stop? Let's have a bit more, and then that's bad for inequality, right? Like, you can imagine this as an attack vector where people come at you, and I think when you're trying to do big things like he is, he's trying to minimize the number of attack vectors. I mean, not recently. He's I say he for various reasons he's created it, but um, I think once this isn't an attack vector, it's seen as it should be seen, which is getting rid of these evils of aging, which are like all these age-linked disease. Like once it's clear this is a force for good, overwhelmingly a force for good. Of course, it can be used for bad, but overwhelmingly a force for good. I think people like Elon are just very reasonable. They will see that and they will change their opinion. But you'd have to ask him directly. Obviously, that's not easy, right? You can't just walk up to him.
Yeah, we'll ask him in the next episode.
I, I'll I'll hold you. Yeah. I'll hold you to account for that.
Yeah. Uh, Daniel, thank you so much for talking to us today. I feel we've spoken a few times now. I feel like there are other longevity biotech startups that are getting a lot of attention, and I feel like you and your company should get a lot of that attention going forward. I hope that will be the case.
Yeah, so that's the purpose of the manuscript. We're actually gonna, in a scalable way, talk to people about what we've done. So, please share the manuscript, forward it to somebody that's getting interested, doesn't know what's real, can't separate dollars raised from credibility. You know, like that's sort of a proxy for
Yeah.
credibility at the moment. Dollars raised and Nobel Prize winners, but you know, we're trying to show that this is now happened, and the conversation can move on to the exciting bit, right? Which is, let's go, right? Like, let's develop these drugs and let's start doing things which sort of blow your mind if you think about them long enough. But that's that's the reason we're all here.
Thank you, Daniel.
You're very welcome.
Thank you.
You're very welcome. Thanks, Patrick. Thanks, Peter.